Okay, so then I guess I'll get started. I want to thank Mara and Utah State for this opportunity to talk with this group, and you know, I'm very grateful for the wonders of technology that allow me to talk to a group in Utah and still be here in Tucson and be able to share this information and also hear your thoughts and during the discussion part of this, and I always learn a lot by talking with growers and certainly researchers, extension people from other universities. Well, this talk is going to be sort of a walk through time. The actually the earliest parts of this were part of my PhD research when I was at Michigan State University. And what the goal was of that research was to build a model where we could predict cross-pollination and fruit set in apple orchards. And when we began this project, we were brainstorming, okay, what kind of things would we need to put into the model? Well, certainly weather conditions, because they're going to affect how many bees come out of the hive and forage on the apple trees, and how the bloom is going to progress, because, you know, you'll have years where it's cool and bloom can last, you know, almost 2 weeks, and other years where it's real warm and in about a week it's over. So we needed to get weather information into the model along with orchard design, because the arrangement of the trees, we thought at that point was going to be very important to cross-pollination. And also the flower density on the trees was going to drive how much fruit could be set. And then finally, colony number, because that was going to determine the foraging population on the trees and how much cross-pollination was going to occur. So as many of you I know that apples need cross-pollination, where these pollinizer trees supply pollen that the bees then move to the main cultivars. And, you know, at this time when we first started this work, this was our thinking, that this is how cross-pollination occurs. And there was evidence in the literature for this, where the fruit set on these trees that were right next to the pollinizer was greater than those that were further away. And so it made sense that, you know, bees are moving between trees, and that's how cross-pollination goes on. And so these pollinizer trees would be interplanted, rows of them. And so in our orchard design, we were able to capture that by the description of the orchard. So we did all of our equation derivations and set up the model and programmed it and debugged it, and we were good to go. And we put subroutines in there that were able to incorporate hourly weather data from weather files that we could collect. And we made a user interface so that they could you know, to put the orchard design in, what cultivars were in the orchard, how were they arranged. Based on the weather conditions and the orchard design, we were able to predict how many open blossoms were available on each of the cultivars during every hour of the bloom period. Based on the colonies that were put in the orchard, we predicted the foraging population and foraging population on apples based on those predictions of open blossoms. So this was all kind of coming together. We were excited about this, and then we estimated the foragers moving from the pollinizer to the main variety based on the foraging population and how heavily different cultivars are being foraged and how much reward was out there, and the movement of bees after they found unrewarding flowers. We had all this logic and all these equations to capture that. And so we set the model to going, and our fruit set estimates, what we found is we found an apple here and there. We hardly set any fruit, and we didn't set anywhere near the fruit in our model predictions that we were seeing out in the trees. So this was a little disheartening, but although this is the power of building models, because you can think things work one way, and then when you start putting numbers on them, you find out that maybe it's working another way. So we went back to the drawing board to look at what was limiting fruit set in our model. And after going through all of our logic and things that could be limiting fruit set and eliminating them one by one, what we found was when we got to, okay, how big is our population of bees moving between trees in cross-pollinating directions? They had to go between rows of different cultivars to make them truly cross-pollinators. We found that population was really small. And if that was really small, then our amount of fruit that was being set, the cross-pollination of those blossoms, okay, that was going to be really small too. So that's when we realized, we don't know how big this population is, but it's probably bigger than what we're predicting. And we don't really know, I guess, how it's created, if it's much larger than the way we were predicting it. So we thought, okay, what we need to do is let's go sample some bees on apple blossoms and see what proportion of them have cross-pollen on their bodies. And then let's go sample the pistils off of apple blossoms and look at the stigmata of those pistils and see what proportion of those are cross-pollinated. And we could examine these pollens and find which ones were self-pollen and which ones were cross-pollen using scanning electron microscopy. I realize there's other ways to do this now, but we're talking about 1984 and our options were limited. So anyway, what we did was we went into a scanning electron microscopy facility and put pollens that we knew what cultivars they came from. And actually we took pollen samples from all the cultivars that were in the orchard, but also all the plants that were blooming in that orchard. So we had dandelion and we had yellow rocket. There were some plum trees that were blooming, and so we collected it all and took pictures and made this sort of gallery of pictures that then, when we took the pollen off of the bees that were foraging apple blossoms or off of the stigmas of those apple blossoms, we could match it up to our gallery pictures and see what type of pollen it was in terms of self-pollen or cross-pollen. Well, what we found, and we were working with orchards that had Red Delicious and McIntosh, so back in those days in Michigan. These were the two most prominent cultivars. In addition, Gold Delicious was really popular as well. I realize these have changed, but like I say, we're going back in time here for this research. But anyway, well, what we found, the punchline is that all the bees that we looked at had cross-pollen on their bodies, and they also had self-pollen on their bodies as well. This is when they were foraging on apple blossoms. And they also had non-apple pollen on their bodies. So it was like whatever was in bloom in the orchard was on the bees' bodies that were visiting these blossoms. And this is really surprising to us. We didn't expect to see this because honeybees have this foraging strategy called foraging fidelity, where they'll continue to pollinate or visit flowers and pollinate plants that are of the same type and even go back to the same row of apple trees in the case of apple trees, or the same area of apple trees. They're very conservative in their foraging. The majority of the foraging population is not shopping around out there looking for something to forage on. Once they find a rewarding foraging source, like an apple tree that's in bloom, they'll just continue to go back to it throughout their foraging day. So we expected to see very little diversity in the pollen. that was on these bees, and what we found was just the opposite. And this pollen was also getting onto the stigma of these flowers. The stigma, like many of you know, this is the pistil, and up here at the top is the stigma. This is a picture of a stigma from an apple blossom, and you can see that there's all kinds of pollen all over it. And again, the percentage that is cross-pollen is here on the Red Delicious was about 77%, on the Macintosh a little over 90%. And again, these stigma also had non-apple pollen on them. They had self-pollen, so they were, in terms of pollen diversity, were resembling the bees that we examined. So then our next question was, where is this population coming from? If there's foraging fidelity in honeybees, and they're visiting apple trees, how are they getting this pollen from other blooming plants on their bodies? And what are the steps that are making this cross-pollination and making this cross-pollinating population so large? Well, if you're going to look at where all this pollen is, of course, it's in the colony. And a honeybee colony has a foraging population that it sweeps out across the landscape. And part of the foraging population, for example, are visiting apple trees. And another part of the foraging population is visiting that patch of yellow rocket that may be over there on the edge of the orchard. Others are visiting dandelions on the orchard floor. And so there's this diversity of pollen that comes into the colony, which is really important for their nutrition. So the hive was the place to look, with all of this varieties of pollen coming into the hive. We thought maybe they're transferring this pollen in the hive, and that's where this cross-pollinating population is coming from. So to test this, we took bees that we emerged in an incubator, so we knew that they didn't have any pollen on their bodies, and we pinned them at the entrances of hives so that bees coming in and out of the hive were bumping up against them and rubbing up against them. And then we also took some of these pollen-free bees that we made, and we put a little dot on their backs, and we released them into hives. And these hives, as I mentioned, were in apple orchards. So we released these bees and let them run around the hive for the day before we collected them back again. And then, so at the end of the foraging day, we collected the pinned and the bees and the ones we released, and we looked at them for pollen. And then we also took half of them and cross-pollinated apple blossoms that we had bagged throughout the day to see if the pollen that these bees were picking up in the hive, if indeed they were, was viable and could be used to set fruit. Well, did the bees have apple pollen on their bodies? Yeah, they did. They all of them, all of the pinned bees at the door had pollen on their bodies, and portion of it in our first year, about 13% of it was apple pollen. In our second year, we had about 70% of the pollen that was on those bees was apple pollen. In the second year, the bees we released in the colony, these are the free running bees, also had about the same percentages of apple pollen as the ones that were pinned at the door. And we also collected drones from these hives. These are male bees, and they don't visit any flowers. And we thought these would be the purest indicators of pollen that was being transferred among bees in the hive. And we found about 1/5 of the drones had apple pollen on their body. I mean, almost all of them, again, had some pollen on their bodies. So was the pollen viable? When we hand pollinated our apple blossoms, we had about 93% set. When we hand pollinated with the pinned bees, we were almost 50% of the apple blossoms that we hand pollinated with the pollen from pinned bees set fruit. In the second year, again, we had about almost 80% of our hand pollinated set fruit, but about 80% of our pinned bees also set fruit. So there was not only a fair amount of apple pollen that must have been coming in, but it had high viability, and that, that makes sense. That's fresh pollen that's coming in from the field, and so that collection of bees that occurs at the door of colonies that are vigorously foraging, there's pollen being exchanged among them, and it's fresh stuff that's coming in, and apparently has high viability. The free-running bees that were in the colony all day picking up pollen, just a little more than a third of those could set fruit, but still, fair amount of viability of that pollen that's in the hive. And the drones, about 22% of those drones that we hand pollinated blossoms with, those blossoms set fruit. So they also were picking up viable pollen. So we reprogrammed for in-hive pollen transfer in the hive, and our fruit set predictions improved tremendously because our cross-pollinating population increased greatly. And we came to realize just how valuable honeybees are in pollinating crops such as apples or cherries, certainly almonds, and because of this in-hive pollen transfer mechanism where the bees don't have to move from tree to tree, but can move this pollen around in the hive and create this foraging population. And it's due to all of this physical contact that goes on the frames of bees, and as they find food inside the colony. Here it's a little bit of honey that dropped on the frame, and you can see the ball circling around and eating that honey, and they're crawling over each other, and there's just a great deal of physical contact that goes on that causes this pollen to be moved around. But in-hive pollen transfers, it's not a foolproof system. It is only as good as the foraging weather. When bees are foraging, it's warm outside, sunny, not a lot of wind, in-hive pollen transfer can really get revved up because there's a lot of fresh pollen coming in, and there's a lot of action at the door and of the colony, and pollen is being moved around. And this can cause fruit set actually in things like apple orchards to get set very quickly. But it depends on the foraging population size. That strong, healthy colonies are essential to have a large foraging population size to again really work in favor of having a lot of pollen coming in, getting moved around, and then going out the door on this population of cross-pollinating bees and cross-pollinating blossoms. Bloom overlap as we would run simulations with this was so important, because you can have great foraging weather, a huge foraging population, but if the proportion of pollen coming in, let's say, is from a main cultivar and very little pollenizer pollen is coming in, what you'll see is heavy fruit set on the pollenizer and lower fruit set on the main cultivar. We would see this, for example, in Michigan, where they were using Golden Delicious as pollenizer for Red Delicious. Golden Delicious bloomed after Red Delicious, and you could never set the king bloom on a Red Delicious. And it was, because when this in-hive pollen transfer got going, the bees were primarily foraging on Golden Delicious trees. And it wasn't until the Red Delicious came out, and so there was, they were foraging on the Red Delicious trees. It wasn't until the Golden Delicious came out that we had compatible pollen. So we didn't set the king blossom on Red Delicious, but we sure set it on the Golden Delicious. As a matter of fact, the Golden Delicious was overset. So this mixture of pollen in the hive is extremely important in terms of having in-hive pollen transfer working in your favor. So, as I mentioned before, the foraging population size is really the foundation of getting in-hive pollen transfer to work. And there are factors that limit foraging population size that have to do, of course, with the weather conditions, colony number that's introduced into an orchard. But even if you have great weather and lots of colonies, the next hurdle to getting a large foraging population is colony health. And so if I could stop now and see if there's, you know, anything in the chat or anything anyone wants to talk about, I'll stop sharing here.
Okay, so far there's been no questions that came
Okay.
through in chat. But we can give a second and see if anyone has any. I know there's a whole mix of people in the audience, from backyard to orchardists to people from USDA here. So if anyone has a question so far, you can type it in. So I know you're working with Washington State University and putting an actual model on their decision aid system. Is that right?
That's the ultimate goal of this. We have a forage model on there now, and we're working on that apple model. And so that's ultimately what we would like to do, is to have growers know when, you know, fruit has set in their orchard, so that if they want to, you know, take the bees out of the orchard because maybe they need to get into crop protection, that would be fine, but also to avoid overset. And the goal that we set was this precision pollination, where we could get bees in and out. Well, actually, I shouldn't say get bees in and out, because another thing they do is hand thin. So they want to know that window when, yeah, my crop is set and the rest of these blossoms are overset, and I want to start taking them off. And you can, I'm sure, understand why this is a delicate operation, because if they're not fully set, you've taken off your crop. If they're set too light and you take them off, now you have apples that are way too big, right? And we've all seen those in the store. So it's you're trying to find that window and predicting it accurately for different orchard designs under different weather conditions is something that certainly modeling can do, but that's ultimately what kind of decision support we'd like to give growers.
Nice. Well, some questions have come in.
Okay.
So let me start at the first one. Do you recommend pollen inserts to enhance in-hive cross-pollen transfer?
Yes, absolutely. I'm glad you mentioned that, especially depending upon your combination of cultivars in your orchard. If your main cultivar blooms first, then you may want to use pollen inserts until your pollinizers come into bloom. The thing with pollen inserts is you don't need them for very long. If you just want to set the king bloom on your trees, then you put them in when your king bloom is open, and when the king bloom is done, and especially if you've got really good weather and lots of bees, the king bloom is boy, that's most of your crop, right? And so you can probably feel good about that your crop is set, and you don't need to put the pollen inserts in, because I know that pollen can be expensive.
Okay, good. Does the presence, or I'm not sure if you can answer this or address this question, does the presence or absence of bees impact apple trees that have gone into biennial bearing?
That's where oversetting is kind of your worst nightmare, and some cultivars are much worse at this than others, because if you overset this year, then your flowering is going to be reduced next year, right? And so especially if you overset and don't thin early enough. If you thin early enough, you're going to be fine. But if you thin after what's called, I believe it's bud initiation stage, it's about 6 weeks, 4 to 6 weeks after bloom, then you won't get return bloom. So, having bees in the orchard for perhaps too long a period of time, or, just setting too much fruit will cause that biennial bearing.
Okay, good, thanks. So when your research, when you collected samples of pollen from those bees, did you release the bees afterward?
No.
Okay.
They were in little bottles in the freezer.
Okay. Have you experimented with horizontal hives and the effect in creating larger hives for larger forager populations?
Oh, that's pretty cool. No, I've never done anything with horizontal hives.
These are good, some good questions.
Yeah, they are.
So this question is just, when you're talking about bee health in a few moments, are you going to mention anything about chemical pesticides or no? This was
just a little bit, and there's a lot to talk about there.
Yeah.
And we can certainly talk about it because we do some work with this, and particularly with things like fungicides that are used in orchards. And but what I was going to talk about in terms of the health was Varroa.
Okay. And one last question. So the issue with the early apples not getting proper pollination, and I know this isn't your area of expertise, but they're saying, well, can you utilize the blue orchard bees for that timing? So kind of have a dual pollination system.
Yeah, I mean, blue orchard bees, can work really well in apple orchards. What drives it with in-hive pollen transfer, but also blue orchard bees, is if there's cross pollen out there for them.
Okay.
So if they bloom real early, but they're all the same cultivar, all blooming early, and nothing's blooming for days, then yeah, it won't matter if it's a honeybee or a blue orchard bee. They're foraging. They're trying, but they're not setting fruit.
Well, that covers the questions. Thanks everyone for those great questions.
Okay, so let me. So now we're going to talk for a little bit about colony health, and this is a real challenge for beekeeping. Beekeeping used to be kind of an easy thing to do, as long as you managed the bees, you kept them fed, put them in a nice location. It was really hard to kill a colony of bees, but now it's really hard to keep colonies alive. And why is that? Well, we seem to have a lot fewer flowering plants than we used to. And anybody who's kept bees for any length of time really sees this decrease in flowering plants and the availability of flowering plants. Although we have protein supplements for bees that you can feed them when pollen's not available, or you can feed them sugar syrup and such, they're not a substitute for pollen and for nectar, which have a lot of nutritional qualities to them that aren't captured in these supplemental feeds. And these nutrients are essential for solid immune responses in bees, and increasing brood production in bees, so colonies will grow. And so there's really no substitute for flowering plants. Another problem is pesticide exposure, especially growers in certain areas. When I was in Michigan, I remember we used to say during the spring when everything's in bloom, everybody wants beekeepers' bees to set orchard crops and so forth. And when all of that pollination was done, it was hard for beekeepers to find safe places to put their colonies, because now all these crops are set and they need protection. I mean, it's, between fungicides and pesticides, applications, bees had a really hard time finding safe spaces to be. And as we know that these sublethal exposures of pesticides have multiple repercussions on honeybees, and can ultimately cause the demise of a colony. Not always, but in certain situations they can. Pests and pathogens, there's diseases out there, and they'll also take out honeybee colonies. And once they get in your orchard or get into your apiaries, they can be a real challenge to control. But the number one problem with trying to keep honeybees alive right now is this little guy over here, these Varroa mites, parasitic mite that lives on the outside of bees. And When they parasitize the bees, shortens their lifespan. These mites transmit viruses. And if you have large mite populations going into the fall, that colony probably won't make it through the winter. And just to tell you a little bit about the Varroa life cycle, how these guys operate, this is our queen bee, and she lays an egg. And 3 days later, the egg becomes a larvae, and the larvae is being taken care of by this nurse bee. It's feeding her secretions from her hypopharyngeal glands up here in her head and helping this little larvae to grow. But you notice this nurse bee also has a little Varroa mite in them. These Varroa mites are really attracted to these nurse bees. These nurse bees put out a particular odor so that the Varroa use these odors as a means to find nurse bees amongst all the other bees in the colony. So here's this nurse bee with a Varroa on it and feeding this larvae to keep, the larvae begins to grow. And just before now this larvae is sealed, that little Varroa will jump off that bee and climb down into this cell and hide down here in the bottom, and the cell gets sealed. And now we've got a developing bee with a parasite in the cell. And this is called a mother mite, and she's mated. And what she'll do is she'll start feeding on this developing pupae, and she'll lay an egg, and the first egg will be a male. And then she'll lay a second egg, and the second egg will be a female, and the male and the female will mate. And meanwhile, this mother mite will keep feeding on this pupae and keeping what is the parasite's wound open in this bee, so it insect blood can clot, and that prevents it from clotting because she continues to feed on it. And her offspring will come and feed on this poor little bee, and it will then emerge. Here's the male. Male will stay down there, and he'll die in the cell. And here's the mother mite and her daughter, and they'll crawl out of the cell. Sometimes the mother mite can make up to 2 daughter mites. If this was a cell that was a drone, because drones take longer to develop than worker bees do, it'll definitely have 2 of these daughter mites along with the mother mite coming out of this cell. And so, then the life cycle starts again, because these young bees that come out of their cells, the first job they have in the colony is as nurse bees. And the mites are right there. So they got this all worked out here in the brood area of the colony. And this population of mites will continue to parasitize then new brood. And as I was mentioning, one of during this feeding, viruses are transferred, and one of the worst viruses that are transferred by Varroa is the deformed wing virus. And actually, deformed wing virus is kind of interesting because we used to find deformed wing virus in colonies all the time, but it was asymptomatic. I mean, you never saw bees with deformed wings or anything, but if you tested bees, you would find, during certain times, their low level of deformed wing virus, but it was never a problem until it got into the mix with Varroa. And now bees with high enough titers of deformed wing virus will emerge, and if I find my arrow here, we'll see full-blown symptoms of deformed wings. And of course, this poor little bee isn't going to live very long in the colony. And if you've got a colony that has a lot of Varroa in it, particularly as you go into the fall, you see a lot of these bees with deformed wings, and that colony's not going to make it through the winter. I mean, it doesn't even matter if you go in there with a miticide, say I'm going to kill all these Varroa that are in the colony. You have a majority of the adult population going into the winter with virus that's going to reduce their lifespan, whether they're showing symptoms or not. And colonies that have populations going into the winter that have reduced lifespan in their adult worker bees will either die over the winter or shortly after in the spring when they begin to forage again. That's the bad news about Varroa mite. What used to be the good news about Varroa mite was that their population growth was really slow. As I said, in worker brood, a mother mite will, in most cases, just replace herself. So that's flat population growth. Or she'll reproduce a second time, but usually they don't make more than, say, 2 mites per mother mite. And while you can get population growth, of course, with that, if you start off low, it takes really some time before you get into large numbers of mites in your colony. This is an output from another model that I built that is a colony population dynamics model that tracks population growth, and we put a little module on it so we can track Varroa population growth right along with the colony population growth. So this red line is the total mite population in the colony. This is mites that are on the bees, but also in the cells. And these free running mites, these are just the mites that are on the adult workers that are in the colony. And you can see if you start off early with, in the year, with low populations, by the time, you get into the fall, you've got maybe, in this case, we predicted about 150, 160 mites in the colony. And, if this colony has, let's say, 30,000 bees, and you got about 150 mites in there, it's going to be a little difficult to even find mites in that colony. And so what we would tell beekeepers to do in terms of keeping mites under control was to, measure this mite population as you went into early or mid-August, early September, and hit them with a miticide at that point in time and drop that population down. And you only needed 1 miticide treatment a year, and you could keep that population down. The mites or the bees would go through the winter and begin to build again in the spring, and whatever mites were left in the colony would build again with them. And then the next year, you would again time that miticide treatment for mid-August, early September, and you were in good shape. You had mites, but they weren't killing colonies. But boy, have things changed. Currently, Varroa population growth looks like this, where you start off with these low number of mites, but they increase throughout the year, particularly in the fall, until in the fall, in this group of colonies that we measured, we were up here about 13 mites per 100 bees in November. And these colonies, this is actual data, this isn't model predictions. This is thousands of bees in this colony, or thousands of mites in this colony that started off with, less than 100 in a single year. And so to keep this population down, you need to start putting miticides in here and possibly moving forward. Well, I can tell you that, well, I don't want to get ahead of myself in my story, but this population here is very difficult to control. But to walk this back, where in the heck are all these mites coming from? Because you can't account for them with mite reproduction alone. So we went and looked at the literature. I mean, sort of a rule of population dynamics is that if you have population growth that exceeds reproductive rates, a place to look for an explanation is migration. And when we looked at the literature for incidences of Varroa mite migration in colonies, we found that it was being documented quite a bit. It wasn't being quantified, but it was being documented as happening. And we didn't know how much it contributed to the mite population, but we knew that people had documented mites migrating into colonies on foragers. So what we decided was to measure migration rates in colonies. And the way that we did this was we altered the entrances of honeybee colonies. And fit these PVC tubes into them. So the bees were coming in and out of these tubes that we stuck here at the entrance of the colony. We blocked off this entrance here, so we just had this hole. And these PVC tubes had a little slit in them that we could drop this screen gate, so that this part of it would capture bees coming in from the field, and this part of it, you can see it's in the colony entrance, would capture bees that were leaving the colony. So we could capture foragers coming in and going out and determine the proportion that had Varroa mite on their bodies. So what we do is take a measurement, let the bees forage for a given number of seconds, put a little cork on the bees coming in here at the entrance part of it, take this out of the colony and put a cork on this end, and then have our 2 samples of incoming and outgoing foragers, and then measure the number of mites we had on those foragers. What we found, this is just one data set, was that foragers with mites were rare in the summertime, but began to increase once we got into the fall, and really increased as we got into October and November. If we look at the relationship between finding foragers with mites and mite population growth in those same colonies, what we found was that as the percentage of foragers with mites are increasing, this is where we go into this sharp increase in mites per 100 bees in the colony. And we've been collecting data sets. This is from 2016. We've been collecting data sets looking at this for several years, and this seems least wise in Arizona and in our apiaries here to be a trend that we can replicate year after year. We see this in commercial apiaries as well, and in commercial apiaries, we see these trends on steroids, where it is the mite numbers are overwhelming, and they're moving around in these large apiaries. And when colonies are collapsing, those mites that are from those colonies are collapsing, either from a loss of a queen or some other problem, but the mites are dispersing on foragers, entering other colonies, causing spikes in mite populations. And so what's happening now is that commercial beekeepers are applying a lot more miticides, and need to keep them on, particularly in the fall, or these bees that are going into winter aren't going to have a chance of making it, because they're bees that are comprising winter population. If they're parasitized and have high virus titers, and as I mentioned before, reduced lifespan, when you take them out in the spring, that population is going to drop, and it may drop to a point that it can't build up again. This is a particular problem with beekeepers who are moving colonies into almonds, where those bees will start foraging again in February, and they don't have a lot of brood in them, and so bees that are dying aren't being replaced, and populations then decline in colonies, and you can end up losing colonies. And the cause of it was what was going on the previous fall with these Varroa mites. So they've made it much more difficult and much more expensive, because you need to use miticides for commercial beekeepers, and it's a real challenge to keep bees alive with this going on in the environment. So that leads me to our latest research area, is why don't we get these bees out of the environment, particularly as we go into the fall, where this Varroa mite migration is starting to rev up, and you're just going to need to continue to sample and possibly continue to keep miticides on your colony to keep them safe before they stop foraging for the year. If we could take these colonies and put them into cold storage facilities, then we've shut down mite migration in these colonies. And another reason to put colonies in cold storage is that in the fall, if you don't have adequate pollen, then you're feeding these colonies. And as weather continues to get warmer, particularly in the fall, bees are foraging longer. They can be foraging in some areas on into November. And so they're burning up stores. Depending upon the latitude you're in, they may not be making a lot of brood. And so they're requiring nutrients to forage or to continue to forage. They're not rearing brood. Mites can be migrating whenever bees are foraging, particularly in the fall. We decided to get into the research area of overwintering bees in cold storage to try and alleviate these problems. And others are also looking at overwintering bees in cold storage. So we have some collaborations with other researchers on this. And beekeepers now are increasingly looking to put bees in cold storage. So our research areas are looking at when's the best time to put the colonies in cold storage, and which colonies are the ones you want to put into cold storage, because not all colonies do well in cold storage. And they have to be healthy colonies that you put in. And they also have to contain what's called winter bees. And we found this out in a study that we did a year and a half ago now, where we had colonies that were in North Dakota and had shut off brood rearing pretty much, and we put them in cold storage. We put one group in cold storage in October and another group in cold storage in November. And the beekeeper also had bees in Texas and wanted to put those bees in cold storage too. Now, the bees that were in North Dakota, we felt pretty good that they had winter bees. And the reason we were saying that is that they were taking in resources and not rearing brood. So they were storing those resources within their own bodies. And they need to be able to do that if they're going to make it through the winter in a cold storage facility where they're not able to get out of the hive and they're in this tight cluster, thermoregulating, staying active, and not rearing brood until sometime in late January or early February. Those bees down in Texas were still rearing brood when we put them into the cold storage facility. And the difference in the way that those two colonies looked when we took them out in February were like night and day. The ones that came from, that were in Texas were smaller, few of them made it through in cold storage. And we feel that they weren't winter bees. They didn't have the resources in the bees to be able to make it through cold storage. So if beekeepers in more southern latitudes want to overwinter in cold storage, we're going to have to develop management schemes, particularly for that. And to do that, we're going to need to understand this formation of winter bees a little better. Our colonies that we overwintered in cold storage, we had more than 80% of them come out of cold storage that were large enough to use for almond pollination. But when we looked at the populations of those colonies, we lost bees in cold storage. Those, colonies went in a lot, I shouldn't say a lot larger, but somewhat larger than they came out. And so we're now asking if we would optimize nutrition and maybe manage nosema a little better in those colonies, whether we could have colonies coming out of cold storage facilities with a lot more bees in them. So we're sort of fine tuning that now and looking to develop recommendations for beekeepers so that they can reduce their colony losses by putting bees in cold storage and have colonies available for early blooming crops like almonds or other orchard crops where there's a reliable source of healthy bees.